This video is about the cellular and molecular basis of cardiac pumping. Topics include the electrophysiological basis of the cardiac action potential, which is what triggers the heartbeat, and how that action potential is coupled to calcium signaling, and then how that calcium triggers contraction. So first, a quick review. The cardiac cycle. So hopefully you've seen this before. The muscles of the heart contract during systole. That makes the pressures in the chambers increase. So when left ventricular pressure, which is here in blue, gets high enough to open the aortic valve, then the blood is ejected from the ventricle, increasing aortic pressure here. At some point, the ventricle relaxes, ventricular pressure drops. During this relaxed phase, the diastolic phase, the ventricle fills again here, okay, and the cycle repeats with ejection and refilling and ejection again, etc. Okay, so what we're going to do is we're going to dig down into the cellular processes underlying these phenomena. It's interesting to start by looking at some of the cellular and molecular structures that make up the heart. So here's a heart. Let's say size, let's say on the order of 10 centimeters. Most of the tissue of the organ is made up of muscle cells called cardiomyocytes, heart muscle cells. Inside a cardiomyocyte, you'll find bundles of proteins arranged in parallel. The bundles are called myofibrils. You'll find lots of mitochondria. In fact, the heart is about 1/3 by weight mitochondria. So this picture underestimates the amount of space that mitochondria should actually be taking up. If you watched our video on myocardial blood flow, we pay some attention to oxygen demand and oxygen delivery. And this is why. These guys here, these mitochondria. So they need oxygen to do their jobs. In particular, they have the big job of making ATP so that, for example, these proteins here in the myofibrils can do their jobs. And here's a zoom in on those proteins and an even closer zoom. At the end of the video, we'll review how these proteins called myofilaments work to generate tension in the wall of the heart to drive contraction. Now, I want to impress on you the size scales here. It's really kind of fun. So going from centimeters to microns here. That's a 10,000-fold difference in size. So to give us some perspective, think about a house. A 2-story house is about 6 meters high. Okay, something that's 10,000 times smaller than that is 0.6 millimeters. So that's about the width of a pencil lead. Right? So if your heart were the size of a house, these few bundles of myofibrils would be like the size of a pencil lead. So now zooming down to one of these individual actin filaments, we have another 1,000-fold size difference, right? So remember, if your heart's the size of a house, a bundle of a few of these myofibrils are like the size of a pencil lead. These individual filaments that are responsible for generating the force of contraction are like 0.6 microns in diameter in this thought exercise. So how big is that? That's something like 1/1,000th of the width, sorry, 1/100th of the width of a human hair. Okay, so again, the heart's the size of a house. One of these little filaments would still be much too small to see with the naked eye. So I want everybody to start with a pretty solid grounding in basic electrophysiology. You might recall that under resting, non-excited conditions, a muscle cell has a negative electrical potential on the inside compared to the outside. That negative potential is due to an excess of positively charged potassium ions. Now, on the face of it, that sounds counterintuitive and kind of confusing, right? So if you're confused, that's okay. Let's just take a minute to get things straight. Look at this picture. There's an excess of positive potassium ions on the inside. That means that there's a concentration gradient driving potassium out. And these potassiums are going to diffuse out until an electrical gradient is set up to oppose and to balance the concentration gradient. So if the membrane is leaky to potassium ions in equilibrium, there's an electric field pushing them back in. That electric field is associated with a negative inside compared to a positive outside. So if there were no potential difference, then that concentration-driven diffusion of potassium would represent a current of positive charges out of the cell. And that current is what sets up the electrical potential difference. So when things are in balance, you are in equilibrium. And there's a famous equation to explain that equilibrium. It's called the Nernst equation. And how do we get the Nernst equation? So the first thing you need to know is a convention. Okay, so the convention is that the membrane electrical potential is defined as inside minus outside. Okay, and you have to be careful to keep track of the signs if you want to follow this. So if the potential is is inside minus outside, then a positive potential would drive positive charges out. Okay, so here what we're writing down is the driving force into the cell. Okay, the driving force into the cell, that's proportional to the negative of the potential difference, right? So this thing F is a constant called Faraday's constant. The diffusion force driving potassium out of the cell is given by this expression. So it has this log term, and you can see that if the concentration gradient is higher inside than outside, then this ratio is bigger than 1, then the driving force out by diffusion is positive. Okay? And you can manipulate that expression, and you can get an equation for what is the potential when the electrical and the diffusive forces are in balance or in equilibrium. So that's the Nernst potential, also called the equilibrium potential. So just again, what that means is that's the electrical potential that you get when diffusion is balanced by the electrical field force. Okay, and so for potassium, the equilibrium potential works out to be around minus 90 millivolts, minus 90 inside compared to the outside. So that's a little bit of electrophysiology math for you. If you don't care for math, then here's what you really ought to know to have an understanding of what's happening here. First, under resting conditions, potassium is near equilibrium. The negative potential is balanced by the concentration imbalance. Okay, and that's because the resting membrane potential is kind of near the minus 85 millivolts. Okay, and we'll see that in a minute. So the situation for sodium is just about the opposite. Sodium is low in the cell and high outside the cell. That gives a positive Nernst potential. So this means that if the cell were to go to around plus 65 millivolts, that's the equilibrium potential for sodium, then the sodium would be in equilibrium. But it never does that. So sodium is always wanting to go inside the cell. Okay, it's driven by both the concentration gradient, okay, high outside, low inside, and in the resting cell by the electrical force, right? Because the electrical force has a negative potential inside, so sodium is being driven in by both electric field and by a concentration field. So now let's apply those concepts to looking at the action potential of a ventricular myocyte. It looks something like this. So before an action potential is triggered, the cell lives at around minus 80 millivolts, okay, right? Remember, pretty close to the potassium equilibrium. Then something happens, and you get this rapid depolarization. Right here, okay, followed by a weird little bump, then a kind of a plateauish kind of phase, and then a repolarization back to the resting potential, and the whole thing takes around 300 milliseconds. So we number the phases of the action potential 0 through 4. So phase 0 is this rapid depolarization. So what's responsible for that depolarization? Well, something, and we'll get to it, tells sodium channels on the cell membrane to open up and let sodium in. And remember, sodium really wants to come in. So there's a fast inward current called the fast sodium current. Okay? And just about as fast as they open, those sodium channels close again. All right? And so the next thing to happen, phase 1 here, is a transient potassium current that brings things back down a little bit. So remember, potassium wants to go out to bring the cell to its equilibrium potential, which is way down here. Okay, that transient potassium current is not enough to repolarize because it shuts off, and you get this plateau because during this phase 2, there are two things happening. One is the calcium current into the cell, okay, and there's a slow potassium current out. Okay, so now for a while, these two currents are roughly in balance, and that's why things stay kind of flat. But eventually, those calcium channels close, and the outward potassium current starts to win out, okay? And if the only current that's happening is potassium, then that's going to tend to move things back toward their equilibrium potential, back here, back to the resting state. So the time courses of those currents are considered here. So here, the inward sodium current is plotted as negative, okay? But remember, what's happening is that this is a positive charges moving in, and that's what's making the membrane potential go up, okay? This little transient outward current, which gives us the phase 1, is called ITO. I is shorthand for current, TO, transient outward. The calcium current is mostly from L-type calcium channels, so it's called ICAL, or ICAL. That's the other inward current, in addition to the fast sodiums. The inward calcium current is balanced by these potassium currents called IKS and IKR in the phase 2 plateau. When the L-type calcium current, the ICal, shuts off, the major part of repolarization is from the IKR current. Okay, and finally, during the resting phase 4, the membrane potential is maintained primarily through a current called IK1. The concept of a refractory period is very important. It's going to be really important in the study of arrhythmias, for example. So to understand refractory periods, we have to understand how action potentials get triggered or initiated. So they're initiated because the fast sodium channels are voltage gated. That means that they sense the voltage. Specifically, when the cell depolarizes a little bit, the channels sense that depolarization, open up, and then the cell depolarizes a lot. So the initiation of the action potentials is the opening of the so-called fast sodium channels, right, to depolarize the cell. Okay, and then once they're closed or inactivated, they stay inactivated for a while, a couple of 100 milliseconds. That period is called the absolute refractory period. Okay, during that time, you cannot initiate another action potential because the sodium channels won't open. Okay, so there's some other things going on in this picture. First one is the effective refractory period. And by the way, this is all adapted from the Lilly textbook of heart disease. So during this effective refractory period, you can get a little something, okay, if you stimulate the cell, but you can't get enough sodium current to depolarize enough to get an action potential that can actually propagate to throughout the myocardium. Okay, there's also a thing called the relative refractory period. If you stimulate in this period, okay, so the sodium channels have recovered enough to give you a weaker than normal action potential. Okay, and finally, something counterintuitive, there is this thing called the supranormal period, and what happens there is that even a weaker than normal stimulus can trigger an action potential. Of course, in the heart, when everything's working normally physiologically, action potentials aren't being triggered until well after all of these refractory periods have ended. These refractory periods, like I said, are going to be important when we think about certain pathophysiologies and associated arrhythmias. Throughout the action potential, under normal conditions, the concentrations of sodium and potassium are maintained. That is, high potassium inside, low potassium outside, also have high sodium outside and low inside. So if sodium is coming in during depolarization and there's an outward potassium current to drive repolarization, how are these gradients maintained? So we have a series of pumps and exchangers at work. Okay, so for the membrane potential, the biggie is the sodium-potassium ATPase. It moves 3 sodium ions out for every 2 potassiums it moves in. That requires free energy input because first, it's moving those things against their concentration gradients. That is, the pump is building up those concentration gradients. So secondly, remember, the inside of the cell is negative, at least in diastole. So the overall charge transfer is positive out because we have 3 going out to 2 going in. So this pump works against the electrical gradient as well. And we're going to talk later a bit more detail about calcium handling. For the moment, appreciate that there are 2 main ways that calcium is cleared from the cytosol during diastole. Most of the calcium is taken up into the sarcoplasmic reticulum via the sarcoplasmic reticulum calcium ATPase, or SERCA for short. The second path is to exchange the calcium with sodium. So usually this sodium-calcium exchanger, the NCX, operates with 3 sodiums coming in for 1 calcium going out. So that's another 3 for 2 charge exchange. But this thing's not an ATPase pump. It's a passive exchanger. And as long as the membrane potential is not depolarized too much, this thing operates in the direction of removing calcium. Okay? But diastolic calcium is pretty low inside the cell, so the calcium gradient is similar to the sodium gradient. When the cell is depolarized, for this thing to work, and what I'm calling the positive direction, that depends on the sodium gradient. So if you were to inhibit how the sodium-potassium ATPase works, then you would affect how this sodium-calcium exchanger works. Okay, and you might keep that in mind for when you study the pharmacology of these pumps and transporters. Okay. Finally, there's a plasma membrane calcium ATPase. Even though it's in this picture, it's really not important for the cardiomyocyte, so you don't have to worry about it for the heart. And I also want to point out these gap junctions. So, those are specific proteins that connect neighboring cells together to allow for ions to move through. So positive ions move through these cells. Moving through these cells are responsible for spreading an action potential from one cell to the next, and that's how the action potential spreads throughout the myocardium. And that process is illustrated here. So normally a heartbeat is initiated in the sinoatrial node, or the SA node. That's why sometimes we hear the heart rate called the sinus rhythm. The SA node depolarizes autonomously on its own, and we'll see how it does that in a minute. So that depolarization spreads through specialized cardiomyocyte tissue that makes up the conduction system. First, the depolarization spreads to the atrioventricular node, the AV node. It also spreads from these internodal pathways throughout the atria. From the AV node, the depolarization wave travels first down the septum towards the apex, and then it turns back around up toward the base of the heart via these so-called Purkinje fibers. The cells of the conduction system, the internodal pathways, the bundle branches, the Purkinje fibers, all act sort of like nervous tissue, but these are not nerve cells. They are embryonically derived from the same cells as regular myocytes. So they're a kind of cardiomyocyte. Here's a vocabulary word for you, good one to know: dromotropy. It refers to the speed of conduction throughout the system. So a dromotropic agent is one that affects the conduction speed through the AV node. Okay, so that autonomous pacing of the nodal cells. The action potential of the SA nodal cell looks like this, and that's different from the ventricular cardiomyocyte action potential. So what's that all about? So here I'm comparing the action potentials of the nodal and ventricular myocytes. So let's start by looking at phase 0 in both cells. That's the depolarization phase. So the depolarization is slower in the SA cell because it does not have fast sodium channels. So this depolarization here in the nodal cell is due to L-type calcium current. And there's just not as much current here in the nodal cell as there is here in the ventricular. So this little transient outward potassium current, which is apparent in the ventricular cell, is absent in the SA node. Next, the phase 2 plateau doesn't really happen so much in the nodal cell. So that's the responsible channels are still present. It's just that in the nodal cell, by the time the repolarizing potassium currents are turned on, the calcium channels are already closing. Okay, so phase 3 is the repolarization phase. It's present in both cells, basically the same underlying reasons. And phase 4 shows a big, really important difference. So in phase 4, the SA nodal cell is continuously depolarizing. So the ventricular cell, in contrast, is just sitting here, pretty stable at minus 85 millivolts, waiting for a signal. It's waiting for someone to tell it to do something. But the nodal cell is moving on its own. Its electrical potential is moving on its own. And when it becomes depolarized enough, then bang, here, it triggers an action potential. So the current that's responsible for that autonomous depolarization in phase 4 is called funny current, or IF. And that's basically how the SA node does its thing. All right. Now, also physiologically, we want to be able to slow that thing down and to speed it up when necessary, right? So how does that work? So in order to slow things down, the cell is hyperpolarized here on the left. So and that occurs by upregulating potassium channels. So that brings the minimum potential more negative, more hyperpolarized, closer to the potassium equilibrium potential. And the result is that it takes longer to depolarize to reach that threshold needed to open up the calcium channels to trigger an action potential. Okay. There's also a G protein coupled pathway triggered by parasympathetic outflow to the AV node that reduces conduction velocity. So now, so that's the parasympathetic side. On the sympathetic side, we want to speed things up, and that's done largely through stimulatory G protein coupled beta receptors. The resulting increase in cyclic AMP activates the funny current. So you're triggering action potentials faster, and you're also increasing the conduction velocity. And the last part of our story is how the depolarization triggers contraction. Okay, and that's through calcium. So here's a plot of the action potential, the cytosolic calcium concentration, and the tension delivered in a ventricular myocyte during a heartbeat. So the depolarization triggers the calcium release, the calcium triggers tension development, the drop in calcium at the end of the cycle signals relaxation. And there's a name for this whole process. It's called excitation-contraction coupling. Here's the excitation, here's the contraction. And the coupling is through calcium, right? Now, under resting conditions, calcium is really low in the cytoplasm, maybe 100 nanomolar. The free calcium concentration outside the cell and inside the SR are on the order of a millimolar or two. So that's a 10,000-fold concentration difference. Okay, so remember that big calcium gradient is maintained by the NCX and the circa. And now let's walk through the whole process. So the first thing is that a drop in polarization, a depolarization signals voltage-gated L-type calcium channels to open. Okay, you know that those channels contribute to this part of the action potential down here, but they also bring calcium into the cell. So those L-type calcium channels tend to live right near things called ryanodine receptors on the SR. So those ryanodine receptors are calcium-activated calcium channels. So a little bit of calcium from the L-type current leads to a lot more calcium coming out of the SR. Okay, and that whole process is called calcium-induced calcium release. Okay, so calcium goes up, like here. Okay, that calcium binds to myofilaments, causing contraction, right? But after not too long, those channels, the L-type channels and the ryanodine receptors, close on their own. They become temporarily inactivated after opening. Okay, and the circa and the NCX work to remove that calcium, and so calcium decreases here. Okay, so with decreasing calcium, calcium unbinds from the myofilaments, and you'll have diastole, relaxation. A little reminder on exactly what calcium is doing with those myofilaments. So those proteins are arranged in a parallel fashion in the myofibrils. If we zoom in on one of those actin thin and myosin thick filaments, here in this diagram, calcium is present. Okay, and when calcium is present, there are binding sites on the actin filament for myosin cross bridges to attach. Those cross bridges generate force, which tends to slide these filaments across one another. Okay, and as long as enough calcium is around, those cross bridges can keep detaching, reattaching, and ratcheting to generate force and sliding. And that whole process, as you can see, involves binding of ATP, hydrolysis of ATP, and unbinding of the hydrolysis products ADP and phosphate. So ATP is the chemical energy source for the contraction, right? And so when the calcium goes away, these binding sites become inaccessible, and so the muscle relaxes. And finally, very important in cardiology, the Frank-Starling effect. So remember, the Frank-Starling mechanism says that the higher the preload, the greater the filling during diastole, right? The greater the contraction and ejection. So how does that work at the cellular level? So frankly, that's one of those things that we really don't know all the details on, but we do know that if you take cells and measure isometric force as a function of calcium, force goes up with calcium, and that's what these data show. But they also show that force goes up more and at lower calcium concentrations for the higher sarcomere lengths, okay, the more stretch, okay? So this is the cellular Frank-Starling mechanism illustrated in this data set. So this experiment on the left is done at sort of steady, constant cellular calcium. On the right, we're looking at the twitch that comes from a single action potential, again at different sarcomere lengths. So increasing sarcomere lengths leads to much more force. Okay, so the story here is that the sensitivity of the myofilaments to activation by calcium increases with sarcomere length. That's the Frank-Starling mechanism at the cellular level.